Welcome back, cells and tissues. Continuing with bone, we're going to talk about the microscopic anatomy of bone. Again, if you're not subscribed, hit the subscribe button, hit the little bell notification so you know when these come out, when more videos come out. And we're going to jump right in. Got a lot to get through in the 30-minute video. So when we talk about the microscopic anatomy, we're going to be talking about all of the major cell types of bone, which come from the same stem cell. So we have osteogenic cells, osteoblasts, osteocytes, and osteoclasts. The osteogenic cells, these are the progenitor cells, the osteoprogenitor cells, these stem cells. And when they are stimulated, they can change into other cells. Okay, they can change into the osteoblasts, into the osteoclasts. And others remain the same. They maintain their stem cells because once they change, they can't go backwards. So they maintain their position as a stem cell to multiply and replicate. So that way we always have the stem cell availability. Osteoblasts. These are our bone builder cells. The way that they build is that they produce unmineralized bone matrix, which we call osteoid. Okay, so osteoblasts make osteoid. Osteoid is made up of type 1 collagen as well as ground substance. And those are the main key points that we want to talk about with those. As well, osteoblasts, as it's producing the osteoid, some of the osteoblasts get quote-unquote left behind to stay within the bone matrix, so that when the bone matrix gets ossified, the osteoblasts are then converted into osteocytes. Okay, so we have the osteoblasts here. They're producing the matrix. All this stuff here in the yellow is the matrix. This is the ossified portion. And these osteocytes here are staying here. These were osteoblasts at one point. Now the osteocytes, those are the mature bone cells located in structures called lacunae. We are going to talk about lacunae a little bit later when we talk about some of the other structures. Their job is to maintain the bone matrix as well as act as stress or strain sensors. They communicate with the osteoblasts, with the osteoclasts, with the other structures to let everybody know kind of what's going on within the bone, what pressure is happening, what stressor is happening, so that way the bone can appropriately respond. Now, finally, the osteoclasts. The osteoclasts are these big multinucleated cells which are responsible for bone resorption. So they break down the bone, release the calcium back into the bloodstream. Specifically when the osteoclasts are active, so either we're in a state of building bone or we're in a state of breaking down bone. Oftentimes, both are happening at the same time in different parts of the bone, but there's typically one is more common or more prominent than the other, depending on what is going on within the body. And when the osteoclasts are active, as they are, dissolving the bone, breaking the bone apart, it creates these little depressions, these little indents called Hauschup's lacunae. And again, these cells, they have the ruffled borders. This picture right here is the one that I want. You can see the little ruffles, the different shape. It gives it more surface area in order to attach the bone, and it's a phagocyte, so it swallows the material, it breaks it down, it eats it, breaks it down. Okay, now we're going to dive into the cortical bone itself. We touched on this a little bit, where we're going to look at all of the individual structures of cortical bone. So we talked already about the periosteum, the fibrous layer, that osteogenic layer within the periosteum. We're going to talk about there are some layers called lamellae, interstitial and circumferential. We have these structures called osteons, and as well we have canals and these other structures called canaliculi. So we're going to start with the osteon. So the osteon itself is the structural unit of bone. So see these little osteon right here. This is what it looks like. We have a whole bunch of these all put together, all in a row, which helps to make up the bone. Think of it, it's an elongated cylinder running parallel to the long part of the bone. They act as these weight-bearing pillars. So it consists of, it has these ring-like structures. These ring structures are called lamellae. And the lamellae, as it's built in so that the fibers within it are running in different directions. The reason that it does this, I'm not sure if in anatomy you guys have talked about the discs yet, but the discs are the same way, where their fibers or different layers crisscross. And that's because it provides extra strength, and you're able to actually stop forces from many different directions. Rather than just being really, really strong in one direction, you're able to withstand force coming from many different directions. So that's the purpose. You have the different direction of the fibers running. You also have a central canal in the center where the vessels and nerve fibers run. And that is the main points of the osteon, aka Haversian system. You will, for your lab material, you definitely will want to know this picture right here, understanding each of these different components. Now, talking about canals and canaliculi. Again, we have a bunch of AKAs for these things. We have the central canal. The central canal, also known as the Haversian canal, is the one that runs straight through the osteons, straight up and down. And that goes through the core of the osteon, has the blood vessels, has the nerve fibers, give all the nutrients that the bone needs inside. We also have the connecting canals, the perforating, aka Volkmann's, aka transverse. Most often, Volkmann's is the word we'll use on the exam because it's the most old science type name. So these canals are lined with the endosteum, and they go to basically at right angles from the central canal. So they connect the central canals as well as connect the central canals to the inside, to the medullary cavity of the bone. We have another structure within the osteon that is called lacunae. Remember, we've heard that word before. We mentioned it previously. What are they, though? Remember how we talked about the osteoblasts as they're laying down the osteoid, laying down the bone matrix? Sometimes they stay in one spot. They stay within the matrix, and then they come and they get ossification happens around them. These osteoblasts that convert themselves into osteocytes essentially keep themselves like their own little cave, their own little cavern. That is what a lacunae is. So the cell lives within this lacunae. And now we talked about the purpose of the osteocytes is to act as a sensor, to communicate what needs to go on, communicate with other osteocytes, with the osteoblasts, with the osteoclasts. So it makes these little tunnels, these little extra canals, these little hair pencil thin canals that actually connect all of the lacunae together as well to the central canal. So that way it's like a little, if you've ever been at the park, some of the parks will have like the little megaphone speakers looking type things, and you can talk in one, and there's a pipe that goes underground and goes to another one, and you can hear all the way in somewhere else. It's kind of like that, where they have these little tunnels where they can share messages back and forth from each other in order to let other osteocytes, let the other parts of the bone know what's going on and what needs to be done in order to maintain homeostasis and make sure that the bone is healthy and strong. So here we have that osteon. We enlarge a single layer right there. And you can see here the lacunae, the little divot inside or around the osteocyte. And then these little tunnels, which are the canaliculi, connecting the lacuna together. Now, we have the osteons, but what about everything that's around them, right? Because circles don't fit together perfectly. So we have interstitial as well as circumferential lamellae. The interstitial lamellae is just all of the lamellae, all of these little units, the structures of bone, the layers of bone that are in between all of the osteon. They fill in all of that space. And then we have the circumferential lamellae, which we have external and internal circumferential lamellae, which are again just the layers of the bone that run around the outside and the inside of the cortical bone. Talking briefly about clinical application, we'll dive way deeper into all of the clinical application in future courses, specifically like imaging courses, pathology courses, those ones, because we talk about bones themselves, especially like cancer originating in bone. It's not super common that bone is the primary source of the cancer, meaning that the cancer cells are bone cells themselves. Okay? A couple conditions that we'll talk about, and you'll see these over and over and over again, like osteosarcoma, multiple myeloma, chondrosarcoma, Ewing sarcoma. These words might not mean a lot to you right now. In the future, they will. So they're fairly uncommon as far as everything goes. More commonly, you have metastatic tumors. So secondary, the bone is the secondary site. So you have like breast tissue, lung tissue, prostate cancer, kidney cancer, thyroid cancer, any of those kind of cancers, liver cancer, that actually goes and it spreads from that other organ into the bone. So that was all the cortical bone. Now, spongy bone, just a little bit information inside it. So it appears very poorly organized and random, but just understand it's organized with specific lines of stress to help the bone resist those outside forces, which we talked about. As well, it's like the cables on a suspension bridge. There are no osteons within spongy bone. The capillaries in the endosteum supply the nutrients, and surrounding the trabeculae is marrow. But just give me one moment. Now, chemical composition of bone, there are organic and inorganic components. So, or the organic side, you have the different cells, you have the osteoid. The inorganic component, the one you really want to remember, is these calcium hydroxyapatites, these mineral salts made up of calcium, made up of phosphate, these crystals. That's what actually goes into the bone, causing it to ossify and to be bone. Now, the development and the ossification of bone. Ossification, also known as osteogenesis, is the process of bone tissue formation. This begins very, very early in utero, so about 6 to 8 weeks-ish. I think this PowerPoint says 8 weeks, but it's 6, 7, 8 weeks within that range that ossification actually starts to occur. So we have here an 8-week, at 8 weeks of development, you have the fibrous membranes and hyaline cartilage of the fetal skeleton. We'll look at both of these, fibrous membranes and then hyaline cartilage, the two different types. These are both going to be replaced with bone tissue through a couple different processes, either known as, there's two different types of ossification. One is endochondral, the other is intramembranous. Endochondral is the more common of the two of them, and it's where originally as a fetus the bones are made up of hyaline cartilage. This hyaline cartilage is ossified and replaced with the cartilage cells. The chondrocytes are replaced with osteocytes through the process of the ossification. Here's this picture in big form. We're going to kind of go through this quickly, as there's just a handful of things that you really need to know from this material, but I do highly recommend at some point, especially if you want to work with children as a chiropractor, that you come back to this and you really understand this process of bone formation, okay? So kind of the first step is you have a development of what is called a primary ossification center. Oftentimes when we talk about like primary, secondary, what you're really saying is primary is the first ossification center. There's the first place where bone starts to form, okay? In long bones, this is typically in the diaphysis, or the shaft of the bone. In irregular bones, usually it's located like in the body of the bone, so like in the vertebrae, within the vertebral body is actually the primary ossification center, with other secondary ossification centers coming up later on. So around that, 8-week mark is his somewhere between 6th and 12th weeks, so about that 8-week mark, you get this formation of a bone collar around what is like around the diaphysis, okay? And this is, kind of cuts to see it, so this would go all the way around, all 360 degrees of the bone. And what this actually does is it cuts off the nutrients to the center, the very central chondrocytes, and those chondrocytes start to ossify because essentially they no longer have the nutrients. They start to die off, and they start to ossify and have minerals and stuff laid down on them. Now this continues. As it continues to grow, you have a deteriorating cartilage matrix that begins to calcify, begins to harden. Finally, kind of in this third step, we get this formation of what's called a periosteal bud, this vascular structure that goes in and initiates kind of replacing that dead cartilage, that broken down cartilage, with actual bone tissue. Okay? The osteoblasts continue to lay down this network of collagen, and the very beginning of this formation, it's not meant to be super nice and neat and proper, because it hasn't had any force or anything put into it to help it maintain in order to help it decide what shape it needs to be in. So we get this disorganized network of bone, or of collagen fibers turning into bone, called woven bone. Woven bone is the first bone that is formed in all of our bones. So the very beginning, it's called woven bone. That is almost always a test question, so be aware of that. Now the bone continues to elongate, continues to grow. It gets longer, like most of these are long bones, and you'll form a medullary cavity, little cavity in the middle where the bone marrow is. Remember that the newborns primarily have all red marrow through all of it. Apologies. So they have the red marrow through the whole bone, and then you get these secondary ossification centers in the epiphysis, okay? So remember for test, primary ossification center, diaphysis, secondary ossification center, epiphysis. Okay? You want to remember that for sure. There we go. Okay. Finally, the last step, you get continued growth, and it gets close to catching up. And we mentioned this before, but you have this physis, this growth plate. This growth plate has a bunch of chondroblasts cells in them. So these chondroblasts lay down cartilage at the same rate as ossification is coming up. So you get cartilage being laid down. This grows from this end. This continues to grow from this end. You get more and more hyaline cartilage being developed. And that's actually how the bones elongate. Eventually, the ossification process catches up to the cartilage process, and it overtakes it, and you get a clothing of the growth plate, and that is when you reach skeletal maturity. Now, the other way is known as intramembranous ossification. So we have, instead of hyaline cartilage, we have fibrous connective tissue membranes, which are formed by mesenchymal cells. And we talked about mesenchymal cells before. They're one of our embryonic cells types that we have. These are they form what are called the membrane bones. So some of the skull bones, like the frontal, the parietal, and the temporal bones, as well as the clavicle and the scapula, are formed through the intramembranous ossification. A lot of our flat bones are more intramembranous. Ones that you'll want to remember, you'll want to remember a lot of these specifically. Remember the clavicle, often tested on. Remember the occipital bone, again, often tested on. And then I would roughly know the others, know which ones are pure intramembranous and which ones also have endochondral ossification. So the way that intramembranous ossification happens is you have your collagen fibers, you have your fibrous connective tissue, and you get this grouping of cells. You get a grouping of the osteoblast cells. And when this group forms, that becomes an ossification center. There really isn't like a primary ossification center in intramembranous. It kind of just happens all over the place all at once, where the osteoblasts, start laying down osteoid. It undergoes its calcification. You get osteocyte, and it continues to expand outwards. So it's a little bit more like a trabeculae kind of different looking pattern. It's more like an inside out kind of approach, and it happens kind of all over, but it gets the job done. Intramembranous ossification isn't tested on as heavily as the endochondral, just because of endochondral is much more common in the body. Just another note about that woven bone. It's that first bone formed by either of the methods. It's remodeled into the mature spongy bone or the compact bone. It's not as strong because it hasn't been fully developed yet. Yeah. Postnatal bone growth. So long bones grow lengthwise by what is known as interstitial or longitudinal growth. They increase their thickness through appositional growth. And then bones stop growing once those physes, once those growth centers have closed. The interstitial bone growth, we talked about, that hyaline cartilage is being put down. When we look at the hyaline cartilage, there are 5 distinct zones. I'm going to fly through these zones. If you have my notes, you can come back and you can read the specifics on them. But more importantly, it's just knowing the name, roughly kind of what's happening in each one, and then being able to identify them is more important than anything. So we have a resting or a quiescent zone. So this is the area of the cartilage on the epiphyseal side of the epiphyseal plate that is relatively inactive. It's just kind of waiting for its time. We have the proliferation or the growth zone. You can imagine what's happening here. The cells are starting to grow. They're going through mitosis. They're replicating. The hypertrophic zone. The cells are enlarging. The cartilage is growing. It's increasing in size. Then we have the calcification zone, where things are starting to get calcified. The chondrocytes are dying, deteriorating to make way for the osteocytes. And then finally, the ossification zone. Jeez, having a hard time saying that word. The ossification zone, where the osteoblasts, osteoclasts are actually coming in and doing their thing. We can see each of these zones right here. Here's another picture. You can kind of see the resting zone, these small just cells hanging out. The proliferation zone, where these are splitting, replicating really, really quickly, pushing this up, growing it. The hypertrophic zone, where these cells are getting much, much bigger. And then the ossification zone right there. There is... I believe it was the proliferation zone, is often referred to as a stack of coins. You can look and see in here, see how the proliferation zone looks like stacks of coins right there. So that is all the development of bone. Now finally, bone remodeling. So this is not just due to injury. Bone remodeling is something that is constantly happening for any number of reasons. Like I mentioned before, it can be because we started doing a new activity. We started weightlifting, we started running. Anything that puts high impact on the bones, we started to introduce new directional forces into our bones. Those bones are then going to need to remodel, restructure in order to better handle the forces that are being presented to them. So it consists of both bone deposit and both resorption. Remodeling also refers to, not to mention, as we grow, this is more that appositional bone growth, growing outwards, right? As we grow, it grows outwards, the medullary cavity needs to expand and get bigger. So we have osteoclasts on the inside eating away at the cortical bone, and osteoblasts on the outside laying down more bone, growing the bone bigger and bigger and bigger, so that the medullary cavity can continue to get wider as well. So here we have. So we have bone deposit, bone resorption, all happening at the same time. You have the osteoblasts, osteoclasts working together for the growth of the bone. Part of the control of remodeling is through hormones. There's two specific hormones. Parathyroid hormone from the parathyroid gland, which is located just behind the thyroid gland, in the posterior aspect of the thyroid gland. Parathyroid hormone stimulates osteoclasts. I really, really want you to remember this, because this is going to come up so many times, especially in the future when we start taking a deeper dive into hormones, into pathologies that involve hormones, as well as like bone issues, which obviously we care about a lot. This is all going to come back a whole bunch. So the parathyroid hormone stimulates osteoclasts. What do osteoclasts do? They break down bone. They release the minerals back into the blood. So what minerals being released? We think calcium. Calcium is the big one. So parathyroid hormone raises blood calcium levels. Another hormone known as calcitonin is produced by these cells known as parafollicular cells, also known as C cells. C cells, calcitonin, the C cells of the thyroid gland, in response to high levels of blood calcium. And this one is going to stimulate the osteoblasts to pull calcium out of the blood and build the bone back up. It's not as big of an effect as the PTH, the parathyroid hormone, but it still does this part of it. So here we have the control of it all. So our blood calcium, because we're using our calcium a lot. In next trimester in physiology, you'll start to see a lot of the things that calcium is used for. Calcium is used for the cardiac cycle in order for the heart to beat. It's used in all of our muscle interactions. It's used in biochemistry. This around the same time, you should be talking about the G protein coupled reactions, and you need calcium through those. When you talk about nerve signals being delivered, you need calcium for that process. So calcium is involved in a lot of different processes. So we really, really need it to be at the appropriate levels. So when we have enough calcium, we're good. If the calcium gets used up and used, it starts to go down. So then our parathyroid hormone activates, stimulates the osteoclasts to bring it back into alignment. Keep our blood calcium nice and level. Here's just an extra little study table. You can look. This kind of focuses on some of those key points, those key histological points that were in this PowerPoint. That was a lot. We made it. Thank you so much for watching the video. Again, make sure you subscribe to the channel. If you have any questions, leave them in a comment down below, and I will get back to them as quickly as I can. Have a wonderful day and good luck studying.